After the structure of plasma membrane and their function, now we are going for the learning of a structure, cell wall, their different modification and their function. Cell wall, we know that is the outermost boundary of a cell, and that is situated outside the plasma membrane. This is the plasma membrane. Yellow lining. This structure is plasma membrane. Outside the plasma membrane, those structure present which is rigid as well as the non-living structure, that is cell wall. Cell wall which is non-living, that's why cell wall which is permeable. It means through which cell wall any molecule can pass. Plasma membrane which is selectively permeable, but cell wall that is totally permeable. Now, what is the function of cell wall? So, cell wall give shape to the cell as well as the protection to the mechanical damages like breaking and bursting. Protection to infection. Cell wall not only give the shape, mechanical damage protection and protection against to infection, also act as a barrier, means obstruction against to entry of undesirable, means unwanted macromolecules, means large size molecule. Large size molecule cannot enter inside the cell because cell wall act as a barrier for the entry of macromolecules. Plus, one cell associated to another cell, cell to cell interaction. So, outermost border, that is cell wall, participate for the interaction between two cells. So, cellular interaction. So, cell wall carry out various functions, maintain the shape of cell, protection against to mechanical damage and infection, barrier against to entry of macromolecules, and cellular interaction. Cell wall present inside one of the plant, that is algae, fungi, as well as rest of the all plant. Cell wall which is made up of specific organic chemicals. For example, cell wall of algae made up of cellulose, mannose, mannans, galactans, as well as the calcium carbonate. Whereas the cell wall of a fungi which is made up of chitin, chitin is also can say fungal cellulose. Fungal cellulose means chitin. Plant, the cell wall which is made up of cellulose is the main component, or in above hemicellulose, pectin as well as protein also take participate. Now most of here organic chemical which is carbohydrate. Now carbohydrate which is three types: monosaccharide, disaccharide as well as the polysaccharide. Most of the component of a cell wall which is made up of carbohydrate, and carbohydrate divide into three types: monosaccharide, disaccharide and polysaccharide. You listen many times glucose. So glucose is the example of monosaccharide. Mannose is also the example of monosaccharide. Two molecule of glucose joint. It means two molecule of monosaccharide join to each other and form what? Disaccharide. Many molecule of glucose means the many molecules of monosaccharide interact to each other and form the polymer structure. So that is can say polysaccharide. So in cell wall components such as cellulose, hemicellulose, pectin and mannans, all these are the example of polysaccharide. Cellulose, hemicellulose, pectin and mannans are the example of polysaccharide. Cellulose is the polymer of glucose. Mannans is the polymer of mannose. Now we are thinking for the further detail about the cell wall. Younger plant cell having a external very thin structure, that is primary cell wall, and that is made up of cellulose. But at the maturity of a plant cell, primary cell wall diminished, reduced and lost. In place of primary cell wall, towards the plasma membrane, toward the side of PM is the plasma membrane, new cell wall developed, which is more thick and rigid. This new cell wall developed towards inside the primary cell wall, towards the plasma membrane. This is secondary cell wall. This is the single layer of secondary cell wall that, like many layers of secondary cell wall, constructed one by one towards the maturity of cell. Here, primary cell wall having ability to grow. Once enough cell size constructed, at that time primary cell wall diminishes. Inside the primary cell wall, secondary cell wall form. This is the secondary cell wall first layer, this is the secondary cell wall second layer. Now till one more layer coming, so that is can say third layer. Symbolically write S1, S means the secondary cell wall. This is S2, now next arriving, that is S3. So these all are, that is secondary cell wall. And secondary cell wall, which is made up of cellulose, organocellulose, hemicellulose, suberin and lignin. Lignin, suberin, hemicellulose, cellulose, that form what? Secondary cell wall. This is another cell. There were also same situation toward the side of plasma membrane. Secondary cell wall developed. Now we are thinking for the association between one cell wall to another cell wall of different two cells. It means two cells glues together, joined together with the help of the structure, that is middle lamella. This is the middle lamella, leads to two plant cells physically joined to each other. This middle lamella, which is made up of the two things, one side calcium and second polysaccharide pectin. Calcium plus pectin, collectively known as calcium pectate. This is middle lamella. This is middle lamella. And this middle lamella, which is made up of calcium pectate. Now one plant cell, cytoplasm, having an association with the nearest plant cell cytoplasm. It means cytoplasm of a two nearest cell communicated to each other with the help of cytoplasmic strand. This cytoplasmic strand that is called plasmodesmata, which is passed through middle lamella as well as the cell wall. This is plasmodesmata. And this plasmodesmata, that is also can say cytoplasmic strand. So two cells communicated, attached to each other with the help of middle lamella, and that is made up of calcium pactate. Two nearest cell cytoplasm associated to each other with the plasmodesmata. In short, chemical changes happen in one cell that is also spread into nearest cell with the help of communicating structure, that is cytoplasmic strand. So now we completely learn here about two cell wall, their structure, their modification, their chemical and the function. Here we have to memorize primary cell wall, which is thin and having a capacity to grow and last. But as a cell mature, so primary cell wall reduce, diminish, and in place of toward the plasma membrane, secondary cell wall develop. Now the next topic, and that is endomembrane system. Endomembranous system. We know clear cut that every organelle having their distinct, means clear cut, structure and function. Every organelle having a clear cut structure and function. But some of the organelle, they are coordinate to carry out the function. It means the function of the different organelle, they are associated to each other, and that form endomembranous system. Examples or organelles including within this endomembranous system, that is endoplasmic reticulum, Golgi complex, lysosome and vacuole. Endoplasmic reticulum synthesis whatever chemical, there were some chemical come in a Golgi complex for the packaging. And though material Golgi complex which is packed, and there is enzyme, so enzyme filling vesicles released from Golgi complex, that is can say lysosome. Vacuole is also associated with these structures. So, endoplasmic reticulum, Golgi complex, lysosome and vacuole all are the component of endomembrane system. But when we think about chloroplast, mitochondria and peroxisome, they are not associated with the above any organelles. That's why chloroplast, mitochondria and peroxisome, they are not a part of endomembranous system. These four organelles with functionally associated to each other. Material synthesized here, coming here for packaging and release as a form of enzyme filling bag, that is lysosome. So they are interlinked to each other. That's why endomembranous system. Our first organelle for the learning, that is endoplasmic reticulum, short form ER. We know clear cut, this is inner nuclear membrane and this is outer nuclear membrane. Between two membrane, the pore structure present, means opening present, through which material communicated between inside the nucleus as well as cytoplasm, that is nuclear pore. So this is nuclear membrane. Nuclear membrane. This is outer membrane, this is inner membrane, and both the membrane form, this is nuclear pore. Nuclear pore, nuclear openings through which material transported between inside the nucleus as well as cytoplasm. Now this green color structure we are drawing here, which is endoplasmic reticulum. Some endoplasmic reticulum we are draw here as a form of flat tiny tubule, and some which is elongated tubule. This flat tube is can say cisternae. Flat tube structure, that is cistern. And now this is pure tubule. What is endoplasmic reticulum? So see, we can see clear cut here. This is the reticulum, is the network spread within the location of cytoplasm. From outer nuclear membrane towards the plasma membrane. So electron microscope observation. Electron microscope observation. According to electron microscope observation within the cytoplasm, the typical complex network or the reticulum of a tiny tubule spread within cytoplasm, that is can say endoplasmic reticulum. So endoplasmic reticulum is the network reticulum of a tubule spread within cytoplasm. This endoplasmic reticulum divide the cell portion inside, so that is can say intracellular space. Endoplasmic reticulum divide entire intracellular space into two compartments. The region inside the endoplasmic reticulum, that is can say luminal region, and the region outside the endoplasmic reticulum, that is can say extraluminal. So endoplasmic reticulum divide intracellular space. Intracellular space means space within a cell into two regions. The region inside the endoplasmic reticulum, that is can say luminal region. Outside region, in between the tubule region, that is can say extra luminal region. Now, this endoplasmic reticulum also divide into two types, rough as well as smooth. So two region of a cell, luminal region as well as the extra luminal region with the help of endoplasmic reticulum. Over the external surface of a cisternae, the organelle attached, and this organelle is ribosome. And we know clear cut ribosome is the site for the protein synthesis. As ribosome present at outer surface of endoplasmic reticulum. That's why this is structurally rough surface. That's why this endoplasmic reticulum is considered rough endoplasmic reticulum. Their external surface, the organelle present, that is ribosome. The tubular endoplasmic reticulum, their external surface having no ribosome. So, that is appear smooth. That's why this endoplasmic reticulum is considered smooth endoplasmic reticulum. SER, smooth endoplasmic reticulum. RER, rough endoplasmic reticulum. Difference between rough and smooth. Ribosome present, ribosome absent. Most of the rough endoplasmic reticulum made up of flat tubule, so there is consistory. Whereas a smooth endoplasmic reticulum, that is made up of simple tubule. Now the function of RER. Rough endoplasmic reticulum synthesize and secrete the protein. It means those cells which is actively synthesize and secrete, means release protein, this cell reach with the rough endoplasmic reticulum. Rough endoplasmic reticulum start from the external outer nuclear membrane and spread towards the plasma membrane. So most of the region of a cell which is covered with the help of this rough endoplasmic reticulum. Now next one, that is smooth endoplasmic reticulum. Smooth endoplasmic reticulum having no ribosome, that secrete the lipid. Animal producing many steroid hormone. Animal producing various categories of steroid hormones. So this steroid hormone is one modification of lipid and which is synthesized from smooth endoplasmic reticulum. So those cell which is actively produce and synthesize the protein, that is rich with the rough endoplasmic reticulum, and smooth endoplasmic reticulum synthesize the lipid, especially which lipid? So animal steroid hormone, that is synthesized from the smooth endoplasmic reticulum. Ribosome associated with the many function. Ribosome spread inside the cytoplasm, that's why it is the component of a cytoskeleton of cell, means along with microtubule, microfilament, intermediate filament. Endoplasmic reticulum provides support to the cell, so cytoskeleton. Within a cell, material transported from one place to another place with the tubule, so intracellular transport is the function. Then we know clear cut. Rough endoplasmic reticulum synthesizes the protein, smooth endoplasmic reticulum synthesizes the steroid or an above glycolipid, as well as smooth endoplasmic reticulum having function detoxification. Any poison, any drugs enter inside cell. So this is neutralized by the activity of smooth endoplasmic reticulum. Formation of organelles such as Golgi complex, microbodies, as well as the lysosome is the combined function of a formation. Lysosome formed by endoplasmic reticulum plus Golgi complex both. Now once again, the intracellular region divided into two parts, inside the endoplasmic reticulum region, so that is luminal region, and outside means cytoplasmic region, that is can say extraluminal region. This is extraluminal region and this is luminal region.